All right, welcome to our lecture on the regulation of gene expression. This information can be found in Chapter 11 in your textbook, mostly Concept 11.3, but there are a few things from other concepts in that chapter, and it tells you at the top of the different slides what concept you can find more information in. All right, so we've already talked about how transcription and translation work. Remember, that's the central dogma. Um, but we haven't really talked very much about how that process is regulated. We've touched on it, but haven't really gotten into too many details before now. So, there are some proteins that individual cells produce all the time. Okay, these are called constitutive proteins because they constitute the normal makeup of what's called the proteome, or the all the proteins together that you normally find within a cell. Okay, and then there are other proteins called inducible that are coded for by inducible genes that are only needed under specific circumstances, right? Only during certain points in your development, or only if the right conditions are met within that cell do they actually need to make this specific inducible um, protein in order to provide some function. So this image shows the central dogma. Okay, remember, transcription reads DNA in order to make RNA. The messenger RNAs are then used as a template in translation, where the ribosome reads the mRNA in order to put together a polypeptide. And then that polypeptide folds up into a functional protein, which carries out the various functions in your cells. Okay, we've already talked at several points about how these can be sort of regulated a little bit. We know, for example, that in transcription, it starts at a promoter, it ends at a terminator. Okay. We know that in the processing of mRNAs, in eukaryotic cells at least, the 3' poly A tail can be extended, which extends the life, because remember, there are those nuclease enzymes that exist in your cytoplasm that are trying to degrade your mRNAs. Okay, DNA is permanent, but mRNAs are not. Okay, similarly, proteins are not permanent either. Okay, and there are various ways for proteins to get tagged for destruction, or tagged for existing for a longer period of time. Okay, but today we're going to talk more about the transcription step and what happens at the promoter and at some other associated sites along the DNA in order to make sure that specific proteins are being transcribed at the correct times. Okay, so we've already talked a little bit, or we've heard this term transcription factor when we did cell signaling. Okay, and often the very last step of a cell signaling pathway is some kind of response. Now, that response doesn't always have to involve the DNA, but if the final response to a cell signaling pathway involves turning genes on, right, meaning making a protein, or turning genes off, which means to stop making a protein, then the very last molecule in that pathway is something that we would call a transcription factor. Okay, and these transcription factors, at least the ones that bind to DNA, are almost always proteins. Okay, so any molecule that can change the expression of a gene either positively, meaning more proteins are made, or negatively, meaning fewer are made, is considered a transcription factor. Okay, and these transcription factors can bind to DNA in various different places. One place that transcription factors always have to bind in order to turn a gene on is at the promoter. Okay, so in eukaryotic cells, again, the promoter is a stretch of DNA. It often contains what's called a core promoter sequence. That is a common sequence found in nearly all of the promoters within your genome. In eukaryotic organisms like us, that is usually going to be what's called a TATA box, okay, which we've talked about before. And having this common core sequence to help identify the promoter of every gene means that you can use common transcription factors to turn every gene on. And we call those common transcription factors general transcription factors. So these are the ones that are going to attach to something like a TATA box, which again is found in basically every promoter within your genome. Okay, there are other transcription factors, though, which are specific to individual genes, and we'll talk a little bit about how that works. This slide talks about some from T cells, right, which means they can only bind to the genes that are important to turn a cell into an active T cell. Okay, so they're probably not, if they're specific to a certain gene, they're probably not going to be binding to the promoter, right? Because again, the promoter has that common core sequence. So there are other places along the genome where these more specific transcription factors can bind. But they work similar to general transcription factors in that when they bind to DNA, both the protein, the transcription factor, and the DNA change shape. Remember, that's that idea of induced fit that we talked about way back when we did enzymes. Okay? Remember, the way that proteins work is by reacting to interactions with other molecules. Okay, so whenever a protein binds something new, there's a chain reaction of bond shifting within its tertiary structure that changes its overall shape. And what that does is either open up new binding sites or hide old binding sites. Okay, so the structure of DNA itself doesn't change in the same way that the protein structure does because of induced fit, but often what transcription factors can do is bend DNA in a way that brings certain sections or segments of your genome closer to each other, or bends it in a way that makes it easier for additional proteins to attach. All right, so this shows the beginning of a typical process for turning a gene on. Okay, so at the top here you see double-stranded DNA. Okay, on the left is the promoter for a certain gene. Inside that promoter is the core promoter sequence, or the TATA box. And then you can see in green there the place where transcription is actually supposed to start. That large brown molecule, TF2D, is a general transcription factor called transcription factor 2D. Okay, it, along with a smaller transcription factor labeled B here, bind to the TATA box and get this process started. Okay, so TF2D and B, right, are going to be used by pretty much every gene in your genome. Because again, every gene in your genome has a promoter, right, which almost always contains a TATA box. So these are general transcription factors. Once they have bound to the DNA, thanks to induced fit, they are now able to bind to additional general transcription factors. And once enough of those transcription factors are bound to the promoter, RNA polymerase, the enzyme which is actually going to make transcription work, is able to bind as well. Okay? So now it seems like we have everything that we would need in order to get this process started, because RNA polymerase is bound, so it can read the template and make the RNA complement. Okay? In reality, though, it's a little more complicated than this. This group of general transcription factors is usually not enough to actually make a gene be expressed. Okay, there are additional transcription factors called activators and repressors that bind to sites on the DNA away from the promoter and away from the gene, sometimes nearby, sometimes very far away in your genome, that also have to be present in order to change how that gene is being expressed. Okay, so you might see on this slide here, some of the text is in red. So the text that's in black and the figures all come directly from your textbook. Okay, this was provided by the people who wrote your textbook. There's an activity that I have assigned for you, though, that comes from HHMI, and it uses some slightly different vocabulary. It uses some additional vocabulary. So, excuse me, any text in red is something that I've added to help you better understand how to do those worksheets. In this case, your textbook doesn't really talk very much about mediators, but mediators are proteins that connect different transcription factors to each other instead of attaching directly to DNA. So E, F, and H would be considered mediators. Okay, and they're going to be really important for connecting this transcription initiation complex that we see here to any activators and repressors that might be bound to DNA at different spots along the genome. All right, so any transcription factor that binds away from the promoter and increases transcription is going to be called an activator. Okay, but any transcription factor that binds away from the promoter but decreases transcription is going to be called a repressor. Okay, so activators and repressors are the proteins, they're the transcription factors, and the places where they connect or attach to the DNA are called enhancers and silencers. So activator proteins attach to enhancer sequences, and repressor proteins attach to silencer sequences. Okay, and depending on which activators are attached to its enhancers and which repressors are attached to their silencers, determines whether or not a gene is actually going to be expressed. Okay, so you have this large complex of TF2D, RNA polymerase, and other transcription factors called the transcription initiation complex. It binds to the promoter. Okay? And then there could be activators or repressors binding to their enhancers and silencers at other parts of the genome. This image shows one of those binding close by, but it doesn't always have to be. Sometimes they can be very, very far away. Okay, so because they prevent transcription when they are around, repressor proteins are involved in what's called negative regulation, while activator proteins, which increase transcription of a gene by their presence, are called positive regulators. Okay. In the worksheets that I assigned for you, both enhancer and silencer sequences are referred to as switches. Okay, so any place on the genome, any sequence where a transcription factor that doesn't bind to the promoter or another transcription factor, right? Remember, the ones that bind to the promoter are called general transcription factors, and the ones that bind to other transcription factors are called mediators. Okay? But any of the ones that bind somewhere away from the promoter, right, the place where they bind in your worksheets are called switches. And the worksheets are going to focus on positive regulation of transcription. So in that worksheet, the switches that they're talking about are enhancers, and the transcription factors that they're talking about, the signal molecules that they're talking about, are generally going to be activators. Okay, so in negative regulation, if the transcription factor is present and it causes transcription to stop, that's what we call a repressor. And the place at which it binds to the DNA is called a silencer sequence. Okay? If the presence of the transcription factor causes transcription to increase, then we say that protein is an activator. Okay? And the place where it binds is called an enhancer. All right, so all of this is really important in terms of coordinating response. Again, what we're looking at here is the end point of a cell signaling process, right? At the very end of many cell signaling processes, transcription factors are produced or released, right? Maybe they had already been produced and they were stored somewhere in a vacuole or something like that, or stored in the Smoothie R or something. Okay, so that last step is going to either create or release previously created transcription factors so that cells can have some kind of coordinated response. Okay, a lot of genes can respond to the same transcription factors. Okay, so what that means is if you need a whole different set of proteins in order to accomplish something, then the genes that code for those proteins have to have the same enhancer sequences and silencer sequences so that they can react to the same activators and repressors. One example of this would be in plants. Okay, a lot of plants have a whole series of reactions that they have to undergo in response to drought. Okay, so all those different genes respond to the same transcription factor that is activated at the end of a cell signaling process in response to drought conditions. Okay, so because the same activator can turn on lots of different genes, then the proteins coded for by those genes are all made at the same time, which is important because you need them to work together to protect the plant during that drought. All right, so that is the information that you should need to help you with the worksheet that I've assigned from HHMI. There's a video to watch, and then you can work on the worksheet. There's a little more information in here, and we'll have some activities on some of this later on. Some of this we've already done activities on. Okay, so the stuff we've already talked about today has to do with regulating genes that are available for transcription, right? So how do you turn genes on and off? By using transcription factors. But that's not the only way that you can regulate this. You can also regulate access to the DNA itself. Okay, and one way you can do that is through a process called epigenetics. Okay, so epigenetics means above the genome. So this is a layer of control that exists above the raw sequence. Okay, and it's one that we now know can be passed on during DNA replication. Okay, so it can be maintained during DNA replication, which means it can be passed on during reproduction. Okay, so as the slide points out, epigenetic changes are not mutations. Okay, mutations are permanent, right? Obviously, they could mutate back again, but once the sequence of the DNA has changed, that is a mutation. Epigenetic changes don't change the sequence of the DNA, they only change the availability of the DNA for transcription. Okay? So, one of the most simple ways to do this is to change a cytosine base into what's called methyl cytosine. Okay, and there are enzymes that do this called DNA methyltransferase. And when you turn it into a methyl cytosine, basically you add a methyl group. Okay? And the methyl group is the carbon and the 3 hydrogens that you can see at the top of the structure on the right. Okay, and we have talked a lot this year about how the shape of molecules influences their function. Okay, and we've talked a lot about how molecules have to have specific shapes in order for them to interact with each other. Okay, so when you add that methyl group to the cytosine to create methyl cytosine, what happens is things like general transcription factors that are supposed to bind to areas of the DNA where the cytosine was cannot bind anymore. Okay, because by adding that carbon and 3 hydrogens, you've changed the shape of the promoter or the enhancer or the silencer enough that the transcription factor protein that was supposed to bind there and turn the gene on can't anymore. Okay, as I mentioned before, this is heritable, and that means both during mitosis in the repair or the growth of your body, but also in meiosis, right, in the production of gametes for reproduction. There are enzymes called maintenance methylases, okay, which read the template strands to see which cytosines have been methylated and then methylate those same cytosines on complement strands during DNA replication. Okay, so this can be passed down. And I'll show you a video I'll post on Friday that's actually really interesting that talks about how this is like epigenetics is sort of the cutting edge. I don't hear as much about it lately, but I know a few years ago everybody was really excited about epigenetics in terms of trying to help us understand some unresolved questions about medicine and inheritance and things like that. So I'll post a video on Friday that you guys can watch along with some questions you can answer. Okay, but the important thing is, even if you pass this methylation or these epigenetic changes down to your kids, okay, again, you are not changing the underlying sequence of the DNA. Okay, so no mutations have happened. And this methylation is not permanent. Okay, just like there is a methylase enzyme that attaches methyl groups, there are demethylase enzymes that remove them again. And if you use a demethylase enzyme to remove those methyl tags, then that DNA goes back to its original shape and is available for transcription factors to attach to again. So that's what you're seeing in this figure here. So you start off with double-stranded DNA at the top. Okay, the original strands are in blue. That DNA gets methylated by a methylase enzyme. When that template is used in order to do replication, the template strands, right, the original ones in blue, are methylated, but the complement strands are not. Okay, but those maintenance methylases that work during DNA replication fix that. Okay, but again, for either one of those new double helices, demethylase enzymes can remove those tags as well and get you right back to where you started. All right, so when a section of DNA has been methylated, as I mentioned before, it is turned off. Okay, so once a gene has been methylated, it basically is unavailable for transcription. Okay, this can be very fine-grained in terms of like one gene at a time, or it can be whole sections of DNA at a time that get turned off this way. Again, because multiple genes often respond to the same switch, right? Whether that's an enhancer or a silencer. And because multiple genes contain similar promoter sequences, the enzymes, the methylase enzymes that put these tags on and the demethylase enzymes that remove these tags can often do this to whole sets of genes, whole coordinated sets of genes at once. Another thing we've talked about is chromatin remodeling. Okay, when we talked about telomeres and chromosomes and things, we mentioned euchromatin and heterochromatin. Okay, and I said that you need euchromatin in order to survive. Okay, because the euchromatin is the parts of your chromosomes where the DNA is relaxed and spread out. Okay, and it needs to be relaxed and spread out in order to be available for transcription. Okay, things like transcription factors can't bind to enhancers and silencers and promoters if they can't access them. Okay, so that's one thing that methylation does. Even though the DNA is relaxed, it can still make those sections of DNA inaccessible. But another way to keep transcription from happening is by turning sections of your chromosomes into heterochromatin. Okay, these are the dark staining areas, and they stain darkly because they are condensed, right? Meaning that they have a lot of histone proteins there around which the DNA molecule is actually wrapped. And when DNA is wrapped tightly around histones and packed together, transcription factors cannot access it in order to start the process of transcription off. Okay, so everybody has chromosomes that are part euchromatin and part heterochromatin. One special case that we also mentioned happens with X inactivation in females. Okay, remember, everybody only needs one X chromosome to live. But every female has 2 X chromosomes in each of their nuclei. So what happens during the process of mitosis is that one of them is randomly deactivated, right? And what that means is that the entire chromosome gets turned into heterochromatin. The chromosome doesn't get destroyed, okay? Because if that cell ever undergoes mitosis again, or if it's a germline cell that needs to undergo meiosis, you need both of those X chromosomes. You need a homologous pair. So instead of destroying one of the X chromosomes, it just gets turned into heterochromatin, right? What's called a bar body. And you can actually see it if you look at a cell. Okay, remember, so this is a female cell in interphase. Okay, you can see the nucleus in there. And remember, in interphase, DNA is supposed to be in chromatin form, right? All relaxed and available for transcription. But to the right of this nucleus, you can see that sort of dark splotch. And that's actually the bar body. Okay, one of this woman's X chromosomes is tightly compacted into a condensed chromosome form even during interphase, right? Normally, you can only see the condensed chromosomes as you move into mitosis. So the process of changing between euchromatin and heterochromatin is called chromatin remodeling. And it's all about how DNA is associated with what are called histone proteins. Okay, so when DNA binds to histone protein, it forms a structure called a nucleosome. And whether or not the proteins, the histone proteins in that nucleosome are acetylated determines how tightly the DNA is going to bind to those histones. Okay, so there are different kinds of histones here. The yellow protein is a histone protein, and the purple ones are too. And the ones that we're interested in are those purple histone proteins in the core of the nucleosome. And we're interested in them because they have that tail. Okay, so each one of those proteins has a tail. The DNA winds around those core histones, and the H1 histone kind of keeps everything in place. Okay, so when acetyl groups are attached to the tail of the histone, it changes, right? Again, remember induced fit. Okay, anytime new molecules interact with a protein, it changes the structure of the tertiary structure of that protein. Okay, so when the acetyl group binds to the tail, it changes the structure of the histones so that the DNA is not so tightly bound, right? The slide here says the histone loses its affinity for DNA, right? That means it doesn't want to bind to the DNA anymore. Okay, so when those tails have acetyl groups attached to them, the DNA gets looser. It goes from heterochromatin to euchromatin, and it becomes available for transcription. Okay, so at the top of this, you see DNA is tightly bound to lots of different histones, right? So I see 6 nucleosomes in the DNA at the top, okay, and they're all pretty close to each other, tightly packed. So at the top here, you have heterochromatin, okay? Histone acetyl transferase enzymes attach acetyl groups to the tails, okay? So those histones become acetylated, and it starts to relax. You can see that those 6 nucleosomes are no longer all that close to each other anymore. Okay, so there are other remodeling proteins that you don't need to know the specifics of, but once it has been loosened, once the DNA has been loosened by by the transfer of acetyl groups to the histones, then these additional proteins can come in and remove histones completely. And that turns the DNA from heterochromatin into euchromatin. Okay, so the third row down, there are only 5 nucleosomes because part of that DNA has been completely unwrapped. There are only 5 nucleosomes. Okay, because one of the histone protein groups has been released. Okay, and because that DNA is now available, things like transcription initiation complexes can now attach, right? And those genes can basically be used. If you look at the top 2 rows, though, this process can also go the other way. Okay, again, the second row is a little more relaxed than the first one because histone acetyl transferase has added histone groups, okay, as you move from the first row to the second, but it can go backwards as well. To go from row 2 back to row 1, you have to remove those acetyl groups from the histone tails. And that is achieved by an enzyme called histone deacetylase. Okay, so when the acetyl group is removed, again, it changes the structure of the histone proteins, making them want to bind more tightly to DNA. Okay, and because they bind more tightly to DNA, that section of the chromosome condenses even more, and transcription is greatly reduced. All right, just a couple more slides to talk about here. The video that I post on Friday about epigenetics will get into this a little more. But a lot of what people are trying to understand now has to do with the old debate of nature versus nurture, right? How much of how you function is determined by the alleles you have, and how much of it is determined by how you're raised, right? Is DNA destiny or not? Or if it's somewhere in between random versus destiny, where exactly do we land in there? Okay, and it turns out that this acetylation and methylation, these epigenetic changes, are probably really, really important in terms of figuring out how you function and understanding why certain things go wrong, right? And understanding how the experiences you have and the environments that you live in change the way that your DNA is used. Okay, so it's not so much that your genome is a blueprint that has to be followed, as it is that your genome is sort of like, I don't know, sort of like an instruction booklet, right? But most people aren't going to use all of it. Okay, bad analogy, but hopefully you kind of understand what I'm saying. Okay, so in that video you'll see they follow some identical twins, a couple different sets of identical twins, right? Where they start off very, very similar as children, but as they get older and experience different things and do different things to their bodies and with their bodies, they start to diverge more and more, both in terms of phenotype and health outcomes. And then what they show in the video is that that's also correlated to the idea that the way they use their genomes gets farther and farther apart the longer they live. The last thing to talk about here is just another way. So most of what we've talked about is about regulating transcription. The last one here is about regulating translation. There are these molecules called microRNAs that can basically bind to messenger RNA. Okay, in order for messenger RNA to work, it needs to be single-stranded. It has to have a start codon and a stop codon. Okay, remember the ribosome attaches at the 5' end. It begins translating at the start codon, and it keeps translating until it hits a stop codon or runs out of mRNA. Okay, but ribosomes can only recognize single-stranded mRNAs. What microRNAs can do is bind to complementary sections of a messenger RNA. And what that does is it makes part of the messenger RNA double-stranded. And if it's double-stranded, the ribosome can't read it, so it effectively shuts off translation of that messenger RNA. Okay, so this is just an example that your book talks about from worms. We don't understand these all that well. Again, this is one of those new things, but a lot of people are looking to try to see if we can use microRNAs as a new sort of class of drugs, right? If your body is producing a protein that is doing you harm, or if you have been infected by a bacterium or a virus that is causing a certain protein to be made, then maybe we could use microRNAs to stop the production of that protein, right? If we can't stop the transcription, with epigenetic changes or with changes to the availability of transcription factors, then maybe we can stop the production of the protein through translation by using something like microRNAs. All right, that's the lecture. So I hope you enjoy it. I hope you're doing well. Enjoy the Stickleback video. And if you want to do those worksheets, just get them to me by noon on Friday, please. I will post new stuff on Friday that will be due by Monday. Thank you. Bye.